Today we talk about proteins. Maybe you are familiar with this word. When we tend to think of a protein as a mass noun, a homogeneous substance, something that your diet should contain in a certain proportion. But if you ever work in a molecular lab, protein may start to look very different to you. How so? Well, you may see firsthand that protein is not just a single substance. There are lots and lots of different proteins in an organism, even in a single cell. They come in every size, shape, and type you can imagine, and each one has a unique and specific job. Some are structural parts, giving cells shape or helping them move. Proteins are among the most abundant organic molecules in living systems and are way more diverse in structure and function than other classes of macromolecules. A single cell can contain thousands of proteins, each with a unique function, although their structure, like their functions, vary greatly. All proteins are made up of one or more chains of amino acid. Protein is found throughout the body, in muscles, bone, skin, hair, and virtually every other body part or tissue. It makes up the enzymes that power many chemical reactions and the hemoglobin that carries oxygen in your blood. At least 10,000 different proteins make you what you are and keep you that way. Protein is made from 20 plus basic building blocks called amino acids. Because we do not store amino acids, our bodies make them in two different ways, each from scratch or by modifying others. Nine amino acids, known as essential amino acid, must come from food. Proteins are highly complex substances that are present in all living organisms. Proteins are of great nutritional value and are directly involved in the chemical processes essential for life. The importance of proteins was recognized by chemists in the early 19th century, including Swedish chemist Jöns Jacob, who in 1838 coined the term protein, a word derived from the Greek "proteus" meaning "holding first place". Proteins are species-specific. The proteins of one species differ from those of another species. They are all organ-specific. For instance, within a single organism, muscle protein differ from those of the brain and liver. A protein molecule is very large compared with molecules of sugar or salt and consists of many amino acids joined together to form long chains, much as beads are arranged on a string. There are about 20 different amino acids that occur naturally in proteins. Proteins of similar functions have similar amino acid composition and sequences. Plants can synthesize all of the amino acids, but animals cannot, even though all of them are essential for life. plants containing inorganic nutrients that provide nitrogen, potassium, and other substances essential for growth, and they utilize the carbon dioxide in the air during the process of photosynthesis to form organic compounds such as carbohydrates. Animals must obtain organic nutrients from outside sources because the protein cannot of most plant is low. Very large amounts of plant material are required by animals, such as cows, that eat only plant material to meet their amino acid requirements. Non-ruminant animals, including humans, obtain proteins principally from animals and other products, for example meat, milk and eggs. The seeds of legumes are increasingly being used to prepare inexpensive protein-rich foods. The protein content of animal organs is usually much higher than that of the blood plasma. Muscles contain about 30% proteins, the liver 20 to 30%, and red blood cells 30% proteins. High percentage of proteins are found in hair, bones and other organs and tissues with a low water content. The quantity of free amino acids and peptides in animal is much smaller than the amount of proteins. Protein molecules are produced in cells by the stepwise alignment of amino acid and are released into the body fluids after synthesis is complete. The high content of protein of some organs does not mean that the importance of the protein is related to their amount in an organism or tissue. Some of the most important proteins, such as enzymes and hormones, occur in extremely small amounts. The importance of proteins is related principally to their functions. All enzymes identify far are proteins. Enzymes, which are a catalyst of all metabolic reactions, enable an organism to build up the chemical substances necessary for life: proteins, nucleic acid, carbohydrates, and lipids, to convert them into other substances and to degrade them. Life without enzymes is not possible. There are several hormones with important regulatory functions. In all vertebrates, the respiratory protein hemoglobin acts as an oxygen carrier in the blood, transporting the oxygen from the lung to the body organs and tissues. A large group of structural proteins maintain and protects the structure of the animal body. All amino acids have the same basic structure: a central carbon atom with a hydrogen, an amino group which is NH2, and an acid group attached to it. Carbon atoms need to form four bonds, so as a fourth attachment is necessary. It is this fourth site that distinguishes each amino acid from the other. Attached to the carbon atom at the fourth bond is a distinct atom, or a group of atoms known as the side group or side chain. The common property of all proteins is that they consist of long chains of alpha amino acids. This is called alpha amino acid because the alpha carbon atom in the molecule carries an amino group The alpha carbon atom also carries a carboxylic group. In acidic solutions where the pH is less than 4, the carboxyl groups combine with hydrogen ions and thus converted into the unchanged form COOH. In alkaline solutions at pH above 9, the ammonium group lose a hydrogen ion and are converted into amino groups. In the pH range between 4 and 8, amino acid carries both a positive and negative charge and therefore do not migrate in an electric field, such as a structure have been designed as dipolar ions or hybrid ions. Although more than 100 amino acids occur in nature, particularly in plants, only 20 types are commonly found in most proteins. In protein molecules, the alpha amino acid are linked to each other by peptide bond between the amino group of one amino acid and the carboxylic group of its neighborhood. The side groups on amino acid vary from one amino acid to the next, making proteins more complex than either carbohydrates or lipids. A polysaccharide starch, for example, may be several thousand units long, but every unit is a glucose molecule, just like all the others. A protein is made up of about 20 different amino acids, each with a different side groups. The simplest amino acid, glycine. Glycine is the simplest amino acid, has a hydrogen atom as its side group. A slightly more complex amino acid, alanine, has an extra carbon with 3 hydrogen atoms. Other amino acids have more complex side groups. Although all amino acids share a common structure, they differ in size, shape, electrical charge, and other characteristics because of differences in their side groups. This is the structure of amino acid. All amino acids have a carbon known as alpha carbon with an amino group, an acid group, a hydrogen, and a side group attached. The side group is a unique chemical structure that differentiates one amino acid from other. The amino acids present in proteins differ from each other in the structure of their side chains. The simplest amino acid is glycine, in which R is a hydrogen atom. In a number of amino acids, R represents straight or branched chain carbon chains. One of these amino acids is alanine, in which R is the methyl group. Valine, leucine, and isoleucine, with longer R groups, complete the alkyl side chain series. The alkyl side chains of these amino acids are nonpolar. This means that they have no affinity for water, but some affinity for each other. Although plants can form all of the alkyl amino acids, animals can synthesize only alanine and glycine. Valine, leucine, and isoleucine must be supplied in the diet. Two amino acids, each containing 3 carbon atoms, are derived from alanine. They are serine and cysteine. Serine contains an alcohol group instead of the methyl group of alanine. And cysteine, contains a mercaptogroup. Animals can synthesize serine, but not cysteine or cysteine. Cysteine occurs in proteins predominantly in its oxidized form. Oxidation in this sense meaning the removal of hydrogen atoms, called cysteine. Cysteine consists of two cysteine molecules linked by the disulfide bond. That results when a hydrogen atom is removed from the mercapto group of each of the cysteines. Disulfide bonds are important in protein structure because they allow the linkage of two different parts of a protein molecules and thus the formation of loops in the otherwise straight chains. Some proteins contains small amount of cysteine with the free sulfhydryl groups. Four amino acid, each consisting of four carbon atoms, occur in proteins. They are aspartic acid, aspargine, threonine and methionine. Aspartic acid and aspargine, which occur in large amounts, can be synthesized by animal. Threonine and methionine can't be synthesized and thus are essential amino acid. They must be supplied in the diet. Most proteins contain only small amounts of methionine. Proteins also contain an amino acid with 5 carbon atoms and a secondary amine in proline, which is a structure with the amino group bonded to the alkyl side chain, forming a ring. Glutamic acid and aspartic acid are dicarboxylic acid. They have two carboxylic groups. Glutamine is similar to aspargine in that both are the amides of their corresponding dicarboxylic acid forms. They have amide group in place of the carboxylic of the side chain. Glutamic acid and glutamine are abundant in most proteins. In plant proteins, they sometimes comprise more than one-third of the amino acid present. Both glutamic acid and glutamine can be synthesized by animals. The amino acid proline and hydroxyproline occurs in large amount in collagen, the proteins of the connective tissues of animal. Proline and hydroxyproline lack free amino group because the amino group is enclosed in a ring structure with the side chain. They cannot exist in a Zwitterian form. Nitrogen containing group of these amino acids can form a peptide bond with the carboxylic group of another amino acid. The bond so formed gives rise to a nick in the peptide chain. The ring structure alters the regular bond angle of normal peptide bonds. Proteins usually are almost neutral molecules. They have neither acidic nor basic properties. This means that acidic carboxylic group of aspartic and glutamic acid are about equal in number to the amino acid with basic side chains. Three such basic amino acid, each containing 6 carbon atoms, occur in proteins. The one with the simplest structure, lysine, is synthesized by the plants but not by the animals. Even some plants have a low lysine content. Arginine is found in all plants. It occurs in high amounts in the strongly basic protamines. The third basic amino acid is histidine. Both arginine and histidine can be synthesized by animals. Histidine is a weaker base than either lysine or arginine. A five-membered ring structure containing two nitrogen atoms in the side chain of histidine act as a buffer, that is, a stabilizer of hydrogen ion concentration by binding hydrogen ions to the nitrogen atoms of the imidazole ring. The remaining amino acid phenylalanine, tyrosine and tryptophan have in common an aromatic structure, that is a benzene ring is present. These three amino acids are essential, and while animals cannot synthesize the benzene ring itself, they can convert phenylalanine to tyrosine. Because these amino acids contain benzene rings, they can absorb ultraviolet light at wavelength between 270 and 290 nanometers. Phenylalanine absorbs very little ultraviolet light. Tyrosine and tryptophan absorb it strongly and are responsible for the absorption band most proteins exhibit at 280 to 290 nanometers. This absorption is often used to determine the quantity of protein present in protein samples. Most proteins contain only the amino acid. Other amino acids occur in proteins in small amounts. For example, the collagen found in connective tissues contain, in addition to the hydroxyproline, small amount of the hydroxylysine. Other proteins contain some monomethyl, dimethyl or trimethyl lysine. Lysine derivatives containing one, two or three methyl groups. The amount of these unusual amino acid in protein rarely exceed 1 to 2% of the total amino acid.
The amino acids are non-essential, meaning that the body can synthesize them for itself. Proteins in foods usually deliver these amino acids, but it is not essential. The body can make any non-essential amino acid, given nitrogen to form the amino group, and fragments from carbohydrates or fat to form the rest of the structure. Some amino acid can be made by the body and are always available, much like the dry ingredients stored in your pantry are always on hand. They are called non-essential amino acid.
Don't let their name fool you. Non-essential amino acids fill essential roles. Non-essential amino acids support tissue growth and repair, immune function, red blood cell formation, and hormone synthesis. Unlike essential amino acids, a healthy body can create these proteins if given enough protein sources with essential amino acid. There are 11 non-essential amino acids: arginine, glutamine, tyrosine, cysteine, glycine, proline, serine, ornithine, alanine, asparagine, and aspartate. There are 9 amino acids that the human body either cannot make at all or cannot make in sufficient quantity to meet its need. These 9 amino acids must be supplied by the diet. They are essential. Your body can't produce or store essential amino acids. It is important to regularly supply your body with these important building blocks. Fill your diet with chicken, eggs, fish, soybeans, nuts, chia seeds, and take a look at the amino acid content of your protein-rich foods. To ensure your body is receiving enough essential amino acids, a good rule of thumb is to consume a minimum of 0.5 grams of protein per pound of lean body weight every day. Older adults, children, and pregnant women might require more protein and should consult with a dietitian for advice. If you are an avid 18 years or older, protein's requirements are much higher and may range between 1.0 and 1.5 grams per pound of lean body weight to optimize recovery and lean muscle growth. The 9 essential amino acids include histidine. Histidine helps make a brain chemical which is neurotransmitter called histamine. Histamine plays an important role in your body's immune function, digestion, sleep functions. Isoleucine is involved with your body's muscles metabolism and immune function. It also helps your body make hemoglobin and regulate energy. Leucine helps your body make protein and growth hormones. It also helps grow and repair muscles tissue, heal wounds, and regulate blood sugar levels. Lysine is involved in the production of hormones and energy. It's also very important for calcium and immune functions. Methionine helps with your body's tissue growth, metabolism, and detoxification. Methionine also helps with the absorption of essential minerals including zinc and selenium. Phenylalanine is needed for the production of your brain's chemical messenger, including dopamine, epinephrine, and norepinephrine. It's also important for the production of other amino acids. Threonine plays an important role in collagen and elastin synthesis. These proteins provide structure to your skin and connective tissues. They also help with forming blood clots, which help prevent bleeding. Threonine plays an important role in fat metabolism and your immune function too. Tryptophan helps maintain your body's correct nitrogen balance. It also helps make a brain chemical called serotonin. Serotonin regulates your mood, appetite, and sleep. Valine is involved in muscle growth, tissue regeneration, and making energy. Your body produces the rest of the 11 amino acids you need. These are called non-essential amino acids. Some non-essential amino acids are classified as conditional essential amino acids. This means that they are only considered essential when you are ill or stressed. An amino acid is an organic chemical. Organic chemicals contain carbon-hydrogen bonds. All amino acids have the same basic structure. Each molecule has a central carbon atom linked together with a basic amino group, a carboxylic acid group or hydrogen atom, and a R group or side chain group. Your body uses amino acids to make the proteins. The different types of amino acids and the way they are put together determine the function of each protein. You don't need to eat foods with amino acid at every meal, but it's important to get a balance of them throughout your day. The recommended daily allowances for every 2.2 pounds of body weight for each of the essential amino acids include requirement of the histidine is 14 milligrams per day and the RDA for isoleucine is 19 milligrams. For leucine it's 42 milligrams and lysine the RDA is 38 milligrams and methionine includes 19 milligrams per day and the RDA for phenylalanine is 33 milligrams. Threonine contains 20 milligrams per day, and the RDA for tryptophan and valine is 5 milligrams to 24 milligrams. Amino acids, which are usually not essential, but in times of illness and stress, may become essential or called conditional essential amino acid. These may be required in conditions such as prematurity in infants. Six conditional amino acids include cysteine, arginine, tyrosine, glutamine, ornithine, glycine, serine, and proline. Experiences periods of extreme trauma or stress from thermal injury, sepsis, surgery, or wounds. Non-essential amino acids become conditionally essential or conditionally indispensable. The physiological demands of recovery may result in the needs for these non-essential amino acids to be greater than the body's ability to produce them. As a result of these demands, the body's supply of non-essential amino acids must be provided by the diet. Arginine, glutamine, and cysteine are examples of conditional essential amino acids that have been combined with other nutrients and supplements formulated for wound healing. L-arginine is 32% nitrogen and have several functions, including accelerating insulin secretion, stimulating protein regeneration, and promoting the transport of amino acid into the cell. Nitric oxide is essential for wound healing because it increases the oxygen and blood flow to the wound, increases collagen formation, and reduces the inflammation. While arginine is the only producer of nitric oxide, 40% of ingested arginine is broken down by the intestines and liver, which reduces the amount available for nitric oxide production. One wound care product is formulated with citrulline, a precursor or arginine. Citrulline circumvents the liver and intestine breakdowns, convert to arginine, and increase the nitric oxide production. Glutamine functions as a fuel source for fibroblast and epithelial cells needed for healing, while the safe maximum dosage for glutamine has been established as 0.57 grams per kg of body weight. Cysteine is required to synthesize glutathione, the cell's major antioxidant that plays a key role during tissue repair and collagen synthesis. It promotes positive nitrogen balance by minimizing muscles breakdown and rebuilding lean body mass. Beta-hydroxy, beta-methylbutyrate is an amino acid metabolite found in foods that slow the protein breakdowns and promotes the production of lean body mass. Lean body mass decreases as we grow. Its preservation is important for healing, especially for the frail elderly. An oral nutritional supplement formulated with glutamine, beta-hydroxy, beta-methylbutyrate, and arginine to increase the collagen formation is marketed for the wound healing. Cells link amino acid end-to-end in a variety of sequences to form thousands of different proteins. A peptide bond unites each amino acid to the next. Condensation reactions connect amino acid, just as combine monosaccharides to form the disaccharides and disaccharides to form the oligosaccharides, and fatty acid with glycerol to form the triglycerides. Two amino acids bound together form a dipeptide. By another such reaction, a third amino acid can be added to the chain to form a tripeptide. An additional amino acid joins the chain, a polypeptide is formed. Most proteins are a few dozens to several hundred amino acids long. Different amino acids can be joined together to make a protein. The order of the amino acids determines which protein will be made. When two amino acids are joined together, a dipeptide is formed. A spherical chemical bond, called a peptide bond, holds together two amino acids. Protein usually consists of multiple amino acids that are held together by peptide bonds. So the bigger the protein, the more amino acids and peptide bonds there are. You know what a protein is. You might be wondering how the amino acids are joined together. A process called dehydration synthesis is used to join amino acid by forming a peptide bond. During this process, a molecule of water is removed in order to synthesize a dipeptide. This water molecule is composed of an oxygen and hydrogen from the carboxyl group of one amino acid and a hydrogen from amino group of the other amino acid. An amide bond joining two amino acids unit is called a peptide bond. Product molecule has a reactive amino group on the left and a reactive carboxylic group on the right. These can react with additional amino acids to lengthen the peptides. This process can continue until thousands of units have joined, resulting in large proteins. A chain consisting of only two amino acid units is called a dipeptide. A chain consisting of three is a tripeptide. By conversion, peptide and protein structures are depicted with the amino acid, whose amino group is free, on the left and the amino acid with a freely carboxylic group to the right. The journal term peptide refers to an amino acid chain of unspecified length. Chains of about 50 amino acids or more are usually called proteins or polypeptides. A protein may be composed of one or more polypeptide chains. For peptides and proteins to be physiologically active, it is not enough that they incorporate certain amounts of specific amino acids. The order or sequence in which the amino acids are connected is also of critical importance. Just as millions of different words are spelled with our 26-letter alphabets, millions of different proteins are made with the 20 common amino acids. Amino acids can be put together in the wrong sequence to produce non-functional proteins. Although the correct sequence is ordinarily of utmost importance, it is not always absolutely required. Just as you can sometimes make sense of incorrectly spelled English words, a protein with a small percentage of incorrect amino acid may continue to function. It rarely functions as well as protein having the correct sequence. There are also instances in which seemingly minor errors of the sequence have disastrous effect. In some people, every molecule of hemoglobin has a single incorrect amino acid unit out of about 300. This minor error is responsible for sickle cell anemia, an inherited condition that usually is fatal. If a person could walk along a carbohydrate molecule like starch, the first stepping stone would be a glucose. The next stepping stone would also be a glucose, and it would be followed by a glucose and yet another glucose. But if a person were to walk along a polypeptide chain, each stepping stone would be one of 20 different amino acids. The first stepping stone might be the amino acid methionine, the second might be an alanine, the third might be a glycine, and the fourth a tryptophan, and so on. Walking along another polypeptide path, a person might step on a phenylalanine, then a valine, and a glutamine. In other words, amino acid sequence within protein vary. The amino acid can act somewhat like the letters in an alphabet. If you had only the letter G, all you could write would be a string of G's. But with 20 different letters available, you could create poems, songs, or novels. Similarly, the 20 amino acids can be linked together in a variety of sequences, even more than are responsible for letters in a word or words in a sentence. The variety of possible sequences for polypeptide chains is tremendous. Amino acids linked together by peptide bonds form a polypeptide chain. One or more polypeptide chains twisted into a 3D shape form a protein. Proteins have complex shapes that include various folds, loops, and curves. Folding in proteins happens spontaneously. Chemical bonding between portions of the polypeptide chains aid in holding the proteins together and giving it its shape. There are two general classes of proteins molecule. First one is globular proteins and second one is fibrous proteins. Globular proteins are compact, soluble, and spherical in shape, while fibrous proteins are typically elongated and insoluble. Globular and fibrous proteins may exhibit one or more or four types of protein structure. The four levels of protein structure are distinguished from one another by the degree of complexity in the polypeptide chain. A single protein molecule may contain one or more of the protein structure types, which is primary structure, secondary, tertiary, and quaternary structure. In primary structure, this primary structure describes the unique order in which amino acids are linked together to form a protein. Proteins are constructed from a set of 20 amino acids. Amino acids have structural properties such as a carbon bonded to the four groups, a hydrogen atom, a carboxylic group, amino group, a variable group. All amino acids have the alpha carbon bonded to a hydrogen atom, carboxylic group, and an amino group. The R group varies among amino acids and determines the differences between these protein monomers. The amino acid sequences of a protein is determined by the information found in the cellular genetic code. The order of amino acid in a polypeptide chain is unique and specific to a particular protein. Altering a single amino acid causing a gene mutation. which most often results in a non-functioning proteins. Secondary structures refers to coiling or folding of a polypeptide chain that gives the protein its shape. There are two types of secondary structures observed in proteins. One type is alpha helix structure. This structure resembles a coiled spring and is secured by hydrogen bonding in the polypeptide chain. The second type of secondary structure in protein is the beta pleated sheet. This structure appears to be folded or pleated and is held together by hydrogen bonding between polypeptide units of the folded chain that lie adjacent to one another. Tertiary structure refers to the comprehensive structure of the polypeptide chain of a protein. There are several types of bonds and forces that hold a protein in its tertiary structure. Hydrophobic interactions greatly contribute to the folding and shaping of a protein. The R group of the amino acid is either hydrophobic or hydrophilic. The amino acids with hydrophilic R groups will seek contact with their aqueous environment, while amino acid with hydrophobic R groups will seek to avoid water and position themselves toward the center of the protein. Hydrogen bonding in the polypeptide chain and between amino acid R groups helps to stabilize protein structure by holding the protein in the shape established by the hydrophobic interactions. Due to protein folding, ionic bonding can occur between the positively and negatively charged R groups that come in close contact with one another. Folding can also result in covalent bonding between the R groups of cysteine amino acid. This type of bonding forms what is called a disulfide bridge. Interactions called van der Waals forces also assist in the stabilization of protein structure. These interactions pertain to the attractive and repulsive forces that occur between molecules that become polarized. These forces contribute to the bonding that occurs between molecules. Quaternary structures refers to the structures of a protein macromolecule formed by interactions between multiple polypeptide chains. Each polypeptide chain is referred to as a subunit. Proteins with quaternary structure may consist of more than one of the same type of protein subunit. They may also be composed of different subunits. Hemoglobin is an example of a protein with this type of the structure. Hemoglobin found in the blood is an iron-containing protein that binds oxygen molecules. It contains 4 subunits, 2 alpha subunits and 2 beta subunits. The three-dimensional shape of a protein is determined by its primary structure. The order of the amino acid establishes a protein structure and specific function. The distinct instructions for the order of amino acids are designated by the genes in a cell. When a cell perceives a need for protein synthesis, the DNA unravels and is transcribed into an RNA copy of the genetic code. This process is called DNA transcription. The RNA copy is then translated to produce a protein. The genetic information in the DNA determines the specific sequence of amino acid and the specific protein that is produced. Proteins are example of one type of biological polymer. Along with proteins, carbohydrates, lipids and nucleic acid constitute the 4 major classes of organic compounds in living cells. Polypeptide chains twist in a variety of complexes, tangled shapes depending on their amino acid sequences. The unique side group of each amino acid gives it characteristics that attract it to or repel it from the surrounding fluid and other amino acids. Some amino acid side groups carry electrical charges that are attracted to water molecules. They are hydrophilic. Other side groups are neutral and are repelled by water. They are hydrophobic. Other side groups are neutral and are repelled by water. They are hydrophobic. As amino acids are strung together to make a polypeptide, the chain folds so that its charged hydrophilic side groups are on the outer surface near water. The neutral hydrophobic groups tuck themselves inside, away from water. The intricate coil shape the polypeptide finally assumes gives it maximum stability. When proteins are subjected to heat, acid, or other conditions that disturb their stability, they undergo denaturation. They uncoil or lose their shapes and consequently their ability to function. Denaturation is irreversible. Familiar examples of denaturation include the hardening of an egg when it is cooked, the curdling of milk when acid is added, and the stiffening of egg whites when they are whipped.